Hello, and thank you so much for joining us today. My name is Wendi Young, and we're going to be talking about pharmaceutical stability. The talk today is going to cover the regulations and guidances for stability, what a stability program entails. We're going to talk about primary stability studies, some supportive stability studies, and then we're going to go into just kind of upper level on data evaluation. I think that could be its own webinar to talk through, but we're going to talk about it just a little bit. So regulations and guidances. The CFR 21 CFR 211-166 has the requirements for stability testing. There are a couple of USP general chapters. There's not a lot in the USP regarding stability testing. There's a lot in the ICH. There's a new draft ICH guidance that is going to make obsolete Q1A, B, C, D, E, and then Q5C for biologics. And we're going to talk, that's primarily what our webinar is today, is going through that draft guidance. 21 CFR Part 211-166 states that you need to have a written program to assess stability, that you need to have an adequate number of batches, which for primary stability is typically 3. You need to have adequate sample size and test intervals to ensure that you are adequately estimating your stability and your shelf life. Your methods need to be reliable, meaningful, and specific, so they need to be stability indicating. Any reconstituted products also have to be assessed, so you need to look at diluent, or if you have a combination product of 2 different APIs that are mixed together, you need to assess that stability. And you need to look at the container closure system that is going to be used for your marketed product. This is the new guidance for ICH. It's stability testing of drug substances and drug products. And again, it combines most of the stability guidance currently in the ICH. It was published on April 11th last year, so almost exactly a year ago. And I think is supposed to go live in November of this year. It has gone through FDA and I think EMA public comment, so we should see adoption by both of those in the coming year as well. So we're going to go through the stability program. The objectives of a stability program, first and foremost, is establishing expiration dating or your retest period for your drug substance and your drug product. You need to use it to confirm any labeled storage conditions. If your storage conditions are room temperature or refrigerated, you're confirming that you are stable with your labeled storage conditions. You're going to demonstrate maintenance of your critical quality attributes throughout shelf life and confirm the suitability of your container closure system. And then stability also allows you to make changes throughout the life cycle, I would say lifestyle, the life cycle of your product. So if you are changing your packaging, then stability is one of the things you utilize to show that it is suitable, that it's not going to have any impact on your drug product or drug substance. In ICH Q1A, they have this figure, and it shows all of the formal stability studies. First one is primary stability studies. These are the studies that you are submitting to the regulatory body to support your application. Then the next is commitment stability studies. If you aren't able to meet all the requirements for stability prior to submission, then sometimes we commit to maybe putting the first production batch up on stability to capture the full scale production batch. Or some other commitment that maybe you need to make to them. Then there's ongoing or annual stability studies. These are just a study that you do. It's just long-term. You don't have to do accelerated. And then product life cycle stability studies, like supporting packaging changes or formulation changes. There are a couple of development stability studies as well. One is stress conditions, and these are temperature and humidity that are more severe than accelerated. And accelerated is 40 degrees Celsius, 75% relative humidity. It's not intended to deliberately degrade your sample, but it's useful in assessing what would happen if you did have an excursion. Would your product still be, would the quality be maintained if there was an excursion? So examples of stress condition studies are thermal cycling and freeze-thaw studies. And then there's force degradation. And with force degradation, you are intending to degrade your product. It's typically heat, light, oxidative, and hydrolysis, so acid and base degradation. And with that, you can investigate what are the degradation pathways, what is your drug product or your drug substance sensitive to with regards to degradation. It also helps you understand the intrinsic stability of your drug product and drug substance. So you can find out if your drug substance is susceptible to light degradation. Maybe that's going to change how you, the conditions during manufacture, things like that. And then force degradation, for us anyway, is primarily used to help demonstrate the stability indicating nature of the analytical methods, making sure that your methods are specific for all the degradants and that they, that you want to make sure that you can actually detect if there is a change that's happening on stability. If you have an assay method and you have degradants that are co-eluting with your API peak, it's going to be really hard for you to know if your drug substance is stable or if you have some degradation happening and you're just not picking it up. So the elements of a stability program, first and foremost, is the approved protocol. The protocol is going to dictate your test, your acceptance criteria, the conditions that you're putting things up at. The batch selection is really important, and we're going to get into that in a lot of detail here in a minute. The container closure and orientation of your packaging. The conditions that you're storing your drug substance and drug product at and your test intervals, your time points, the tests that you're performing, your CQAs, and then the acceptance criteria, your specifications for that. And then once you get all of that done, then once the stability is done, then your data assessment and your reporting, and then any ongoing stability and commitments, including your annual stability. Study design, when you're first starting your stability study, you want to assess any development data that you have. What do you know about your drug substance? What do you know about your drug product? What conditions are you going to store your product at? Is it going to be room temperature? Is it going to be refrigerated or frozen? And then you want to determine what your stability indicating CQAs are, your critical quality attributes. You only need to look at those critical quality attributes that are stability indicating, the ones that could change on stability. There are some critical quality attributes like residual solvents or elemental impurities. Maybe we wouldn't expect those to change on stability. And then you want to assess your analytical procedures for any gaps that you might have. Have you done your force degradation? Is your method specific for all potential degradants? Are there any other methods that you need to develop because you have a stability CQA that you need to measure? And then you draft your stability protocol. Your stability protocol should have the study purpose. Is this primary stability? Is it a development batch? Is it a commitment batch? Packaging change, things like that. What batches are you using and the description for those batches? And then you can put the specification in your stability protocol or have a reference to a published specification. Your container closure system and the orientation that you're going to be storing the batches at. The storage conditions, are you testing at zone 2, zone 4B, accelerated? Are you putting any intermediates up? How are you going to trigger intermediate testing? And then your testing frequency. What are the time points for your study and then study duration? We're going to talk now about primary stability. And primary stability, again, are the lots that you put up to support your application. The standard approach for primary stability is you need to have 3 batches of your drug substance, and then you need to have 3 batches of each strength of your drug product. You need to evaluate accelerated and long-term stability, and all of your drug product container closure systems need to be represented. You don't have to have 3 lots of every drug product container closure system, but you need to make sure that every size is covered in your stability. And you don't have to have your primary stability batches be production batches, but this is one of those commitments. If you don't do that, then you need to have a commitment that says you're going to initiate stability on a production batch. This is a table from ICHQ 1, the proposed guidance, the new proposed guidance. There's 3 different product types on here. One is new synthetic chemical entity. So this is kind of an NCE or an associated drug product. The next is a existing synthetic chemical entity. And then the last one is biological drug substances and drug products. So each of these need to have 3 batches. For the biological drug, you can have smaller batch sizes, but one of those has to be a production size, whereas you have the option to submit it later for the synthetic chemical entity. For the biological, one of those 3 has to be production size. And then the amount of data that you need to have at submission is listed there. For a new synthetic chemical entity, you need to present 12 months of long-term storage and 6 months of accelerated. For an existing chemical entity, it's just 6 months. And then biological is only 6 months. And usually that's because the biologics don't have as long of a shelf life anyway. The critical quality attributes that you want to look at on stability are typically color and appearance. You want to look for phase separation, any changes in the appearance of your drug product or drug substance, content or assay, impurity profile, degradation profile, preservative content. If you have a critical excipient, as an example, we had one that had a biodegradable polymer. We looked at that excipient to track the stability of some CQAs for that. And then microbial limits. The microbial limits typically are only tested at initial and then annually. You don't typically test that on every time point. You want to look at any diluents. You want to treat them as you would your drug product. You want to identify what the CQAs for the diluent are and then test that on stability. And then consider any potential interactions with the container or the delivery devices. And that would be ICHQ3D, which is elemental impurities, and ICHQ3E, extractable leachables. Your analytical procedures need to be able to detect changes in product quality. We said that a few times already. They need to be stability indicating. They need to be validated when you're doing your primary stability. And this is, I think, a new thing in ICHQ 1 is that you need to have a mass balance relationship between the tests. So you can't have if you have like a 5% degradation of a assay, you can't, and you're not seeing that as an increase in your degradation, you're going to need to address that in some fashion. The way that the ICH defines mass balance is that it's adding together the assay value and the levels of the degradation products to see how closely they add up to 100% of the initial value. So you need to kind of take some, you need to take a kind of a scientific lens on that and look and say, okay, with my analytical variability, am I close enough to 100% of my original value? Your stability and your release methods can be different. Your release methods don't have to be stability indicating. And it might be appropriate for you to establish a release specification that is tighter than your stability spec. So if you're expecting a 5 to 10, hopefully not 10, but let's say a 7% loss on stability of your API, then your release specification probably shouldn't be less than 97%, so that you can make sure that you're staying within your stability. And then you need to assess functionality of combination products. Combination products are a drug substance or an API, a drug product, and a medical device. So if you've got a maybe metered dose, you need to assess the functionality, and you need to develop a method or a process for assessing that functionality throughout stability. Selection of your primary stability batches. The drug substance needs to be the same chemical synthetic route. It needs to be a similar manufacturing process of the one that you're going to market with. Your batches can be at pilot scale, and they need to meet the proposed specifications. And then your drug substance need to be in containers that are the same material as your production batches. We'll talk about containers here in a minute. Your drug product needs to be the same formulation and the same dosage form, which is kind of obvious, but you need to have a minimum of 2 batches at pilot scale. Other batches can be smaller. Pilot scale is typically 1/10 of your manufacturing production scale. They need to meet the specification, and they need to have the same fill volume. The fill volume, if you have multiple fill volumes, then you're going to need to put those fill volumes up as well. The same container closure system as proposed for your marketing. So container closure system, drug substance, again, same or representative of the packaging proposed for storage and distribution. You certainly don't want to put 20 containers, 5-kilo containers up on stability. So it needs to be the same type of container and constructed of the same material, but it can be a smaller size. You want to try to, as much as you can, replicate the headspace. For drug product, if possible, you want to use the commercial container closure system. This ensures that the container can protect the drug product, that the container is compatible with the drug product, and that it will function as intended throughout shelf life. Something else that you want to, and one of the reasons that you want to have your commercial container closure is that you need to assess for migration of any packaging components into the product. I give an example of benzophenone as a UV protectant on a label, and having that benzophenone migrate through the label, through the bottle, and into the product. And so because we didn't have the commercial label on stability from our primary batches, we saw this on our first production. And there was a mad scramble to figure out what it was and get methods for it and all of that, and assess the impact, the quality impact of having that in the product. And then orientation, you need to assess the impact of container orientation on your critical quality attributes. For liquids, solutions, semi-solids, and suspensions, you need to place them both in upright and inverted positions. So that is like closure up and closure down. Testing frequency, the time points that you're going to set, if the drug product or your drug substance has a shelf life that's less than 12 months, you need to test monthly for the first 3 months, and then in 3-month intervals after. And again, you're only going to 12 months there. If it's greater than 12 months, you test every 3 months for the first year, every 6 months for the second year, and annually thereafter. And this is detailed in the new guidance. I think this is industry standard, but hasn't necessarily been spelled out like this before. And that's for, both of those are for long term. For accelerated, you need to have a minimum of 3 time points, 0, 3, and 6 months. And if it's likely to be out of specification, you need to increase your testing. So maybe adding a 1 and 2 month time point, so that if you do halt stability, if it's failing at 3 months, you really still need to have those 3 time points. I like to have at least 1 time point between 0 and 3 until you're really familiar with your product. And then you can do a reduced design throughout. Q1, it says that other design can be used, or you can risk assess or provide justification for changes from kind of the standard program. Climatic zones come from the World Health Organization Technical Report 59953, which Annex 2, Appendix 1, I've got a snip of one of the tables here, and this is for the Americas. So you can see Brazil at the top there, which is Zone 4B, and then the United States down there at Zone 2, which is 25C, 60% relative humidity. In the regulation, it says that there's a lot of options for long-term for Zone 1 and Zone 2. You can do the typical 25C, 60% relative humidity. If you do that, then you can trigger an intermediate if your accelerated is failing. But if your long-term is going to be higher temperature, higher humidity, or any of the other zones, there's no intermediate option. The accelerated temperatures for every single zone are the same at 40 degrees Celsius and 75% relative humidity. One of the things that the guidance states is that you can use a more severe condition. So if you are marketing a product globally and you typically have to do Zone 4B and Zone 2, you could, if your product is stable at Zone 4B, you could just do stability at Zone 4B and use that to support a Zone 2 application as well. Refrigerated and frozen conditions. Long-term for refrigerated is 5 degrees Celsius. Accelerated is 25 degrees Celsius. And there's no humidity requirements for refrigerated or frozen. Frozen is less than 20 degrees or negative 20 degrees Celsius. And there's no accelerated requirement for frozen, but it's recommended to do accelerated or some stress condition to help support any excursions. You can imagine that the risk for frozen sample or frozen product excursions is a lot higher than for room temperature. Semi-permeable containers are containers that allow the passage of moisture or solvent or gases into the container, in or out of the container. Impermeable containers provide a permanent barrier to prevent moisture or solvent migration. So those impermeable containers would be like aluminum tubes, aluminum foil blisters, or sealed glass containers. And if it's in an impermeable container, it can be conducted under any humidity condition because there is going to be no transfer of moisture across the packaging. For the semi-permeable containers, any aqueous-based products have to be evaluated for potential water loss under low humidity conditions. So instead of running your long-term stability at 25 degrees Celsius and 60% relative humidity, you're going to run that at 40 degrees relative humidity, and then a 25% relative humidity for your accelerated. If you see more than a 5% loss of moisture in a aqueous-based product that's in a semi-permeable container, that's considered a significant change, and you need to look at your packaging and your formulation. You are able to do reduced testing. We talked above about how you need to represent every strength on stability. It's possible to bracket or matrix your stability design so that you don't have to test every single strength in every single container. So bracketing is when you're testing the extremes only. So you're testing the highest and lowest strength or the largest and smallest filler container. So in the table that's on the bottom of the slide, you can see that there's a 50 milligram, a 75 milligram, and 100 milligram strength of the product, and then 3 container sizes, 15 milliliters, 100, and 500, which is a wild range, 15 to 500. But you can see their bracket that they're doing is that they're testing 3 lots of the 50 milligram at the high and low container size, and 3 lots of the 100 milligram at the high and low container size. So the 75 milligram and the 100 milliliter aren't represented anywhere in stability. The only way you can do this is really understanding that 75 milligram and the 100 milliliter, and knowing that there are no significant differences in the formulation or in the packaging that would negate the ability to bracket. So you need to support your bracketing decision with clear justification, and you have to be able to preserve the ability to evaluate trends and support the shelf life of that 75 milligram and of that 100 milliliter package size. Bracketing is a lot more complicated, and you're looking at different variables like batches, container size, strengths, time points, and you are putting up product that supports all of it, but maybe not testing every single time point. This is really appropriate for large stability programs where you're still able to interpret the stability of everything, and you're able to detect meaningful change, but you're not having to test, maybe 6 samples at every time point. You're just testing 3. In this example, they're testing every single batch, all 3 of the batches for both of the strengths at 0, 12 months, and 36 months. So the beginning at 1 year and at the end of the stability or the shelf life, and then just testing half of those at the other time points. And you can imagine if we added in the other strength into this, if we added in the fill sizes and the container sizes into this, how big this could get very quickly. Now we're going to talk about supportive stability studies. The first one is photostability. Photostability is currently in ICHQ 1B. It's performed to demonstrate that light exposure doesn't result in unacceptable change that could compromise product efficacy or patient safety. The picture right there is of photostability chamber. It's actually the one that we have in our lab. It demonstrates the ability of your primary and or secondary packaging to protect your drug product and drug substance. You only need to look at one representative batch of drug product and drug substance for synthetics. And the data that you generate with your photostability study supports your product labeling and helps you to determine whether or not you need to make any changes or have any precautions in manufacturing to prevent degradation of the product while it's being manufactured. You might need to repeat photostability if you have changes in packaging, if it's not a like-for-like change, or if you have, I think that probably is it, if you've got a change in packaging, if you've already demonstrated that your packaging works. But it's also a big part of force degradation for method development. We look at light degradation. Your photostability study can be done in parallel or it can be done sequential. So sequential photostability study would be looking at your drug substance and or your drug product directly exposed. You don't have it in the packaging at all. And you look at that. If there is no degradation that happens, then you don't need to do any further studying. It's not light sensitive. If you do see some degradation, then you need to look at your drug substance and your drug product in the primary packaging and possibly in the secondary packaging. So your methods really need to be able to discern whether or not there is a non-acceptable change. And this is where the force degradation as a part of method validation is really critical. And if a non-acceptable change, and non-acceptable has to be defined by you, is observed, then you need to change your packaging or you need to change your formulation. Process and hold time. So process and hold time for intermediates is looking at the amount of time that it takes to manufacture a single step or a series of steps, or hold time, how long you can hold your materials. That could be like a bulk hold waiting to package. All of this needs to be done under protocol. You need to do these studies under relevant temperature and humidity conditions. We certainly wouldn't want to hold something refrigerated that we don't intend to hold refrigerated in typical manufacture. And then we need to make sure that light exposure is considered, that you have, that you're using, it's exposed to the amount of light that you would expect that it would be exposed to in a normal situation. And then for any intermediates that are packaged and stored outside of manufacturing, so this would be maybe you have discrete manufacturing steps, and then you hold something for further manufacturing. Maybe it's transported to another facility. You can have an established shelf life or retest period for those rather than having a hold time. Short-term storage is for when the primary packaging is not open, so it hasn't been breached, and you are allowing storage at a different condition. As an example, you have a prescription product that is stored refrigerated, but once it's dispensed to the patient, it has a 3-month room temperature shelf life. So that 3-month shelf life at room temperature shouldn't be applied post package expiration or lot expiration. So if you're evaluating the stability of that, you would want to do it at 21 months so that that short-term storage is concluded prior to expiration of the product. It must be provided as part of the primary stability, but it doesn't have to be repeated unless there's a change that would impact the short-term storage. And then you need to do this on at least 2 batches. End-use stability is a little bit more complicated. It can get very complicated depending on how the product is used. So you need to do stability using conditions that mimic the intended use of the drug product after it has been opened and through the preparation, storage, and administration per the instructions. So this could be a multiple dose drug product, like an oral solution in a single container. It could be a single dose drug product that is handled or prepared and stored prior to administration. So like you're diluting it. Maybe the pharmacist is reconstituting or co-mixing. We had a product that one of the APIs degraded the other. So we had a cream and a solution, and the pharmacist mixed them together. And then there was a shelf life of that was given to the customer. So then you have to establish stability there. The drug product, it could also be the drug product being combined with a medical device for dosing and then how long it's dosed. If there is a drug product that is assembled, it's a combination product with a medical device, and it's used immediately, you don't have to do in-use stability. Your in-use stability protocol, you need to have 2 batches of representative drug product. One of them should be chosen towards the end of its shelf life or from the final point of the stability study. It needs to be commercial packaging, and you can use a bracketing or matrix approach for this as long as you justify it appropriately. And the testing that you do needs to be based on what changes you would expect for in-use. Typically, it is the same testing scheme that is used for stability to assess both the physical, chemical, and then microbial stability. Data evaluation. Again, this is probably a third of the 108 pages of Q1. And we're just kind of have a few slides on it. So data evaluation, the approach for data evaluation, at its core, you're establishing the shelf life or the retest period at the recommended storage condition for your drug substance or your drug product. You want to evaluate trends over time, and you also have to take into account the degree of variability between batches. And that is we talk about poolability, whether or not the different batches that are on stability can be pooled together. And that means that they have a similar trend, and there's not a lot of variability between the 2 batches. And there's limited extrapolation that you can do. And the decision tree here that I've got snipped on there, I know you can't read it, but you can pull it up in Q1. If you look at the first column there, what it says in that first bubble at the top is you need to trend your data. The next one is there significant change at accelerated conditions? And we go down to the next one. Is there the long-term stability? Is there little or no change over time? And is there little or no variability? If you say yes to both, then we ask the same question of accelerated stability, little or no change and little or no variability. If that's the case, then you can extrapolate, and if you have to present 12 months of stability in your application, you can get 2 times that, so 24 months shelf life based on your 12 months of data. So if there's little or no change, little or no variability, you can get double your shelf life of what long-term stability you already have generated. The rest of them goes into, okay, well, if it's slightly variable, and then maybe you only get 1.5 times and things like that. It gets, there's a lot of examples. It talks a lot about models that you can use and things like that. It's quite detailed in the new guidance. So the scope of your data evaluation. Your evaluation needs to be comprehensive. It should include your stored intermediates. What kind of stability do you see during processing? During, what are your process hold times and what does that stability look like? Do you have short-term storage? Do you have in-use stability? Is there a high risk of excursions? As we talked about, frozen stability or frozen storage has a much higher risk of an excursion than a room temperature stability. And an excursion that would have could have a massive impact. So your data evaluation shouldn't be tables and graphs and it's stable for 2 years. It really needs to be an interpretation of all of the data that you have put together regarding the stability of your drug substance or your drug product. So in conclusion, draft ICH Q1 consolidates the stability guidance into a single, much clearer framework for executing stability studies. The core expectations with our current ICH Q1A through Q1E have not changed. You want to make sure that you're aligning your stability design with your intended labeling and your commercial packaging, and ensure that your container closure, your configuration for your container closure, and the orientation are justified. And then you only want to apply bracketing and matrixing when it's scientifically justifiable. And again, throughout the guidance, it states that other approaches can be justified. So if you've got, what I've presented here really is the standard approach. There are a lot of different options that you can take if you need to. And again, you want to present stability study results as an interpretive package, not just a compilation of your data. Thank you so much for joining us today. If you have any questions, please shoot us an email at engage@triveritylabs.com. We'll be happy to answer any questions that you have. We will be having our next webinar in a couple of weeks, and that will be on method validation. Hope to see you there.